Bismillah Rahman Rahim. Welcome, dear student. This is Biochemistry 2 course, code Chem 342. And today we are having lecture 18. Today's topic is zymogens or proenzymes. This is your teacher, Dr. Naeem Khan, Associate Professor, Department of Chemistry, CUST. In today's lecture, we will talk about zymogen or proenzymes in detail. We will highlight different aspects of this concept zymogen. Then we will discuss the activation of zymogen. Actually, zymogen are the inactive forms of enzyme. So we will discuss how these zymogens or proenzymes are activated um, to be fully active enzyme. Then we will move to the mechanisms of the zymogen activation. We will have some examples with the basic steps they follow to be fully active. So let's have a start. Zymogens. Zymogens are among those enzymes. Actually, this is applied to some of the enzymes. Not all enzymes are existing in zymogen. Some of the enzymes, and especially the digestive and coagulative enzymes. Especially the digestive enzymes are the enzymes which are synthesized in the inactive forms. These are the enzymes like digestive enzymes, enzymes required for the digestion of food, which are produced in the inactive forms, means they are not fully active. In the inactive forms of the zymogen, they are called proenzymes or zymogens. So the concept is this zymogen term or proenzyme is applied to the inactive forms of the enzymes which are produced in some cases. Means some of the enzymes like digestive enzymes are not produced as a totally active enzymes, and whenever they are produced in the inactive form, they are called zymogens or proenzymes. Thus, we can say that zymogens are the precursors of enzymes. I mean, these zymogen will give rise to the enzyme, so thus we can say that zymogens are the precursors. They will give rise to the enzyme later on after activation. So we can say that zymogens are the precursors. Generally, the prefix pro or suffix ogen, ogen, is added to the name of the enzyme to denote its zymogen and enzyme. The prefix pro or ogen, suffix ogen. Prefix is something which is added at the start of the name of enzyme. So to designate the zymogen form of the enzyme name is written with the addition of suffix prefix pro or suffix ogen. For example, look, thrombin is an active blood clotting enzyme. When it is produced in the inactive form, it is called prothrombin. This pro prefix designate that prothrombin is the zymogen form of the active thrombin enzyme. In the similar way, proelastase. Actually, the pro designate that this is the inactive form of the elastase enzyme. Similarly, trypsinogen, ogen is the suffix added to the name of trypsin in order to designate the zymogen form of trypsin enzyme. Similarly, pepsinogen ogen is the suffix added to the name of active enzyme pepsin. So this pepsinogen is the inactive form of the pepsin enzyme. In a similar way, fibrinogen is a fibrin protein. Is when it becomes activated, this is called fibrin. the final enzyme involved in the blood clotting mechanism. And whenever it is in inactive form in the blood, it is known as fibrinogen. So, zymogens are something which are represented by adding the prefix pro to the name of enzymes or suffix ogen to the name of enzyme. This will distinguish between the enzyme and its inactive form or proenzymes. Zymogens are the enzyme inactive forms because some enzymes are produced in the inactive form in the body. And the inactive forms are called zymogens or proenzymes. We can say that zymogens are the precursor because they are later on converted to the enzymes. Now, this is a big question that why enzymes are produced in the inactive forms. It is very difficult to argue that enzyme, if precursor form is important, that why not all enzymes are produced, why some of the enzymes are produced as precursors. This is a direct question come to our minds. Why some of the enzymes, not others? As I mentioned in the last slide that some of the enzymes, especially the digestive enzyme, are produced in the inactive form. So the question why? Look, actually, these precursors or the inactive form enzyme production are the evolutionary in our case, are the evolutionary in a corension means survivals. These are actually the from the evolutionary point of view, these were the mechanisms which survives with the time. They are required at times or places. Means these enzymes are required in the specific spots or specific cell in specific time. Means they are not always required. You can say. So the active enzymes would injure the cell. This is a very good argument or answer to this argument. There's actually the enzymes, if produced in inactive form, they will injure the cell. For example, if an enzyme is digesting protein, and if it is produced directly in the active form, it will definitely attack on the cell proteins. We know that cell membrane is made up of protein and lipids. So even if food protein are not available, they will attack on the protein available in the cell. So in this way, the cell will injure. We can say that zymogens production is because of evolutionary, and from the evolutionary mechanisms, these are actually the one which survived. And there are some enzymes. Why some enzymes? Because these are the enzymes which are not always required. They are required at some specific times. And these enzymes, if produced in active form, they will injure the cell. So this is an explanation to the argument that enzymes are produced in inactive form, and the reason is if they are produced, they will injure the cells. Zymogen not active on their own, but they need conversion. If an enzyme is produced in inactive form, zymogen form, so they will not be activated by their own. They need a specific conversion. And what is that conversion? Conversion is actually the biochemical change which is required. What sort of biochemical change? That change is actually the hydrolysis or changing the configuration. We know that in enzymes we have active sites. Until and unless the active site is exposed to the substrate and in a specific three-dimensional configuration, then without that, it cannot be come in contact with the substrate, and in this way the key and lock model cannot be fulfilled. So there is a biochemical change, and that change is actually the changing the configuration. Means changing the configuration, and the idea is to expose the active site in a specific direction so that it suits to the substrate. And usually there is some hydrolysis reaction which are bringing about the change. Look at this example. This is an enzyme inactive form which is protein chain shown, is called and recoiled upon itself. And the activation by hydrolysis of the specific peptide bond, because hydrolysis will result in the breaking of some of the bond. Look this bond here, these bonds are broken, and now we have by hydrolysis some bonds are broken, and this enzyme is now active. So you can say that here a space is provided. So just keep in mind the key and lock model of enzyme. Now substrate can come in contact with the enzyme, and they can give rise to the enzyme substrate complex. This picture is actually from GL Jain Fundamentals of Biochemistry and can be traced there. The activation of zymogen takes place in the Golgi complex, Golgi bodies of the cell, which is the precursor part, and the precursor part is cleaved in order to activate it. Here is some part just in the last slide. We saw that a slight chain is broken down by hydrolysis to activate it. The inactivating piece can be provided, can be a peptide chain or independent folding domain. This means that by the activation mechanism sometimes removes the inactivating piece, which may be the polypeptide chain, and the hydrolysis actually break down their chain. Or sometimes it is an independently folding domain, a full chain that is broken down, and thus the enzyme active points are exposed and the enzyme is activated. Usually, this is up to 100 or more than 100 amino acid residue chain is involved in the inactivation, and that is removed for the activation of enzyme. So in literature reports are there that the inactivated chains are about more than 100 amino acid that is broken down. These are access at the N-terminal extension of the enzyme. We know that in proteins we have the N-terminal or C-terminal. Actually, the amino acids are having two ends, carboxylic end and the amino acid end. And the protein chain formed is actually the amino acid, that is formed in the whole protein chain. The one end is having the NH2 end or amino end, which is called N-terminal, and the other end is having the carboxylic end, carboxylic acid group, and that is C-terminal. So this inactivated group is linked at the NH2 end of the protein, which is going to be removed for the activation of the enzyme. Remember, this N-terminal is actually the one from where protein naming is started, and this N-terminal is actually the start, and we say that actually this is the amino acid number 1 in a polypeptide chain while starting from the N-terminal. This helps in the folding and stabilization of the enzyme. Whenever this strand, the inactivating piece or the precursor part that is linked, what they do, they actually provide stability, and they fold and refold the enzyme. And whenever we remove that, enzyme becomes unstable. We know that from the basic chemistry point of view, whenever a molecule is highly unstable, they will react. A stable molecule will never react. The enzyme is inhibited and the inactive zymogen or proenzyme. Whenever such precursor part up to 100 nucleoamino acids are linked to the N-terminal, they provide stability to the enzyme, and thus the enzyme is inhibited, prevented from its activity, and thus it is termed as zymogen or proenzyme. For example, we have trypsinogen, and this is the proenzyme of trypsin, and it is synthesized by pancreas. It is activated by intestinal enzyme named as enterokinase. Enterokinase enzymes activate trypsinogen, and trypsinogen, ogen. Ogen is actually the suffix for the zymogen. So this coenzyme, this zymogen of the enzyme trypsin is activated by the enterokinase. How? Like this. Look this. Enterokinase is converting trypsinogen into trypsin, and here we can see that we have a chain from the N-terminal. We have valine linked to the 4 aspartic acid residues. Here 4 is written. So this 5, 6 is lysine and 7 is isolysine. What this enzyme enterokinase is doing, it is breaking this down this chain of amino acids of the first 6 amino acids. Look, this valine, aspartic acid, 4 and lysine. For this one, this is 5, this is 6. So a chain of the 6 amino acid is broken down and the remaining chain is now active enzyme. So in order to activate this trypsinogen, this chain must be broken down. Enterokinase is breaking down this, and hence this is converting the inactive trypsinogen into the active enzyme. Next, this figure can be traced in the linear principles of biochemistry, and please go there for a detailed discussion. Another enzyme is the chymotrypsinogen. This is synthesized by the exocrine cells of pancreas. This is a basic, we know, and it is the precursor is in chymotrypsin because ogen is the suffix for the zymogen. So the chymotrypsinogen is the inactive form synthesized for the enzyme trypsinogen. Hydrolysis by is activation is taken place by enzyme trypsin is convert chymotrypsinogen into the active form trypsin, chymotrypsin. How? Like this. So there will be some change brought about by this trypsin enzyme. And what is their change? This is a very good view of the enzyme. Chymotrypsinogen, when produced in the inactive form, there is a chain of 245 amino acids from N-terminal N1 and up to the 4 to 245. Here we can have disulfide linkages between the two cysteine amino acids, and it is at 42 to 58. Also, there is a disulfide bond of 168 and 182. Here, a disulfide bond of amino acid at position number 1, which is cysteine, at the with the amino acid at position 122. And similar way, amino acid at position of 136 is having disulfide linkage with the amino acid at position 201. This is the inactive form when produced. This trypsin bring about some change. How? This break the bond between the amino acid 15 and amino acid 16. Look, this bond is broken, but this the unit is still linked because it is having disulfide bond with the amino acid 121. So this is linked, but this by hydrolysis this bond is broken. No, it is by itself active, and what it does, it break down and remove the two dipeptides, that is serine, and serine is position number 14 and arginine is position number 15. Look here, two amino acids are removed. 14 and 15 are not here because it is 13 and it is 16. So one dipeptide is removed, that is serine at position 14 and arginine is position 15. Another dipeptide is the threonine at 147 and asparagine at 148. Look here, at 147 and 148 is not here because it is 146, 149. And no, this is a fully active enzyme. From this mechanism, we come to know that the inactive form is converted to the active enzyme. How? First by hydrolysis, removing the bond between the 15 and 16, and then by removing two dipeptides, and that is from a position of the 14 and 15 and 147 and 48. By this way, this enzyme is activated. This picture can also be traced in the Zymogen fundamentals, and for any confusion, this can be traced there. Another example is pepsinogen, and is an inactive form of pepsin and is also produced by the gastric juice. Pepsinogen is converted to the active form pepsin both by HCl of the gastric juice and pepsin itself. HCl unfold the pepsinogen. For the activation, this unfolding is important, and the 44 amino acid chain is cleaved by pepsin to activate. The process is called autocatalysis. Whenever an enzyme is activated by itself, it is called autocatalysis. This is pepsinogen conversion to pepsin, and a polypeptide of 14 amino acid will be broken down. and this requires hydrogen ion and also pepsin itself. This autocatalysis of the enzyme means this activation by itself can be represented by a graph. Here, the enzyme activity is shown, and by this way time. With the time, the enzyme activity increases, mean activation occurs. But whenever the enzyme is fully saturated with the substance, then its activity becomes constant. Like this, look, enzyme activity increase, but here it becomes constant because enzyme is fully saturated with the substrate now. And this figure can also be traced in the Jail book of biochemistry. One thing we should not confuse, that enzyme can also occur in inactive form whenever a cofactor is not found. Activation and such enzyme become activated when the cofactor such as coenzyme or prosthetic group is linked. The coenzyme, we know that the protein part of the enzyme is called coenzyme, and that when linked to the coenzyme, which is called as the when linked, it is become activated, and the non-proteinous part and proteinous part when come together, it is called whole enzyme. So the enzyme is become active when it becomes whole enzyme, means when the protein part proenzyme is linked to the non-proteinous part. So this is also a sort of activation, but it is not proenzyme. Enzyme glyceraldehyde 3-phosphate dehydrogenase. This is an enzyme, glyceraldehyde 3-phosphate dehydrogenase. It needs NAD as coenzyme for the reaction. Look, this is a very important reaction of the glycolysis. And here is the oxidation of glyceraldehyde resulting in the formation of the 1,3-biphosphoglycerate. And this enzyme is inactive if NAD is not linked. If this is linked to this enzyme, it becomes activated. So this is another activation, but this cannot be confused with the proenzyme or zymogens. This reaction I took from the Voigt and Voigt fundamentals of biochemistry. Dear student, once again, I referred each and everything to a very textbook. And this today, you people must go to the fundamentals of biochemistry, Voigt and Voigt. It's a very important book. Also, GLJN will be required, especially for the chymotrypsinogen mechanism activation. And Leninger book, that's very important book, can be consulted for the activation mechanism and the introduction of the proenzyme. Please go to these different books and highlight each and every point and develop very good notes. From today, for today, I will say goodbye. Thank you. Allah Hafiz.